Welcome everyone to our second digital meeting. This morning, Channa is going to take us through a lot of the work that he and his colleagues have been doing in Sheffield.
Thank you, Adam, and thank you everyone for joining me today in this sunny Saturday morning. I'm Channa Hewamadduma. I'm an adult neurologist from Sheffield. My specialist interest is neuromuscular disorders and inherited gait disorders such as Hereditary Spastic Paraplegia, which I'm very passionate about. And I'm going to talk to you today about our experience in Sheffield. We have a very large cohort of HSP patients and what work we are doing to help and understand this condition and patients. So these are my disclosures. And so I work in Sheffield Teaching Hospitals Foundation Trust, which is a large tertiary and quaternary referral center, and we cover several CCGs crossing multiple counties, and we cater for about 2.5 million people. So we have HSP patients traveling to our clinic from outside the region, as well as there are some international referrals who visit or connect us with us over video. We are lucky in Sheffield that not only we have the facilities to run an extensive HSP clinic, but we also have access to the cutting-edge neuroscience research, particularly in Sheffield Institute for Translational Neuroscience, which was opened by Her Majesty the Queen in 2010. And last year we were awarded Queen's Award for the neurosciences research we undertake to understand neurodegenerative disorders which affect patients. So under Citron, we bring together clinicians, the scientists, the gene therapists, the computer modelists, bioengineers, everyone working together to understand the mechanisms affecting neurodegenerative conditions, and then also find treatments. When once these treatments are found, that there's a process to seamlessly bring it to the clinic to help the patients. So over the years, there are a number of contributions to HSP from Sheffield. I won't bore you with details, but hereditary spastic paraplegia is not an uncommon term for this audience here, but we all know it is a progressive neurodegenerative condition, and it is very variable, and some patients are very severely affected at a younger age, whereas some might be only mildly affected and they develop the disease later on in their life. This variability is underpinned by different genes which can cause HSP. In this elegant article, we has shown that spastin or spast mutations are the commonest, and thereafter the atlastin, Ki5A, REAP1, SPG11, and the list goes on. Our experience in Sheffield is slightly different to this. This is not a UK cohort, and we know that HSP is numbered according to the chronological order of when the gene or the locus was discovered: SPG1, the LICAM, SPG2, PLP1, and then later on SPG4, which is called SPAST, was identified, but SPG3 was not found for a long time, and when they found it, they called it atlastin. And we know what's happening with HSP patients. They have predominantly spasticity in the legs, and this is due to degeneration of the long motor axons in the spinal cord. Now, why are these long motor axons particularly vulnerable? Because these axons are extremely long. If I were to draw a parallel, that if the nerve body were to be a football, the length of the axon is like 7 football stadiums long. So if the axon is such a long structure, its internal environment needs to be very closely, very tightly monitored and managed. Any deviation from the norm can affect these health of these nerves, and this can start dying. In this spinal cord from a patient with HSP, you can see that this arrow here points towards a part of the spinal cord called the lateral corticospinal tract, which is almost like the M1 of the spinal cord, which takes all important motor functions, signals through this tract. And this has the longest nerves, and then the light color shows that they are degenerating. And over here is the dorsal column, which also takes the sensory signals from your feet up to the brain to tell the brain where your feet and legs are in relation to the space. And these are also quite a long nerves, and they also unfortunately undergo degeneration in HSP. To understand what's actually causing this degeneration, we need to understand various genes that have been discovered in HSP and how they cause impairment in the function of the cell. When you look at the clinical picture of HSP patients, some of them are pure. In other words, they have only spasticity and some degree of sensory changes in the feet, but the others have much more than that. They might have dysarthria, that means slurring of speech or difficulties in walking, and there might be droopy eyelids, or they have difficulties in moving their eyes. They might have cognitive impairment. So to understand how and when somebody might develop these complications, it's important to be able to predict the natural history of this group of patients, and we do not have much information published with this regard. When you look at a cohort of HSP patients, some of them have a family history, but most of them are sporadic. That means they are the first person to be affected. The inheritance of the condition could be autosomal dominant. That means there is a faulty gene that goes from one member to the other, from parent to the child, or autosomal recessive. That means you need to have two abnormal copies. For example, SPG7 or SPG11 are affected in this manner. It is an orphan condition. HSP is an orphan condition because in 2 to 5 people per 100,000, when you look at the worldwide population, are affected. So it is very important that there are HSP support groups to bring awareness and bring together the patients and clinicians, and then also clinicians to become aware of this condition. So what is our cohort in Sheffield? Our HSP clinic has been running for nearly 20 years, initially started by Professor Dame Pamela Shaw and then Professor Christopher McDermott, and I currently lead the HSP service in Sheffield. We have a number of clinicians, gene therapists, physiotherapists, and neuroradiologists and bioengineers working together. We have a database over the years of about 420 HSP patients, and we have a clinical data set available for 371 of them. And more than 200 of these our patients are genetically characterized. That means they have a genetic diagnosis. So this is a large number of patients to be housed in a single center. This brings a lot of advantages of being in one center because these patients have been examined by 1 or 2 clinicians only, and they have a follow-up data over a period of time. And when you look at our data set, more than 50% of these patients are pure HSP patients. Only 47 are complicated. And if I draw your attention to the pie chart below, you could see that nearly 60% of them have spastin mutation. And what is interesting is about a quarter of them, this orange slice, is SPG7 or paraplegic. And we also have a considerable number of SPG31, that is REAP1, SPG11, and K5A and atlastin mutation patients, and a few families with all the other genes which cannot be put in a pie chart here. So what is important to remember is that in our cohort, there is this large group of paraplegic gene mutated patients. And this gene was thought to be rare in British cohorts. And now we are increasingly recognizing this. This might have implications for some patients who don't have a genetic diagnosis yet, because this tends to be causing mild disease in some and tend to be late onset, so patients might not be recognized by clinicians and GPs. So when you look at our patient cohort and analyze their phenotype or in their clinical picture, They seem to be slowly progressing, predominantly symmetrical. That means both sides are affected same, and they have spasticity in their legs. There are some patients who have early onset HSP, and it is interesting to analyze them to find that some of them have been wrongly called cerebral palsy. And when their family member was found to have HSP, these patients were then reclassified as Hereditary Spastic Paraplegia when they were tested for the familial gene. And they also have some minor sensory problems in their feet, which might contribute to their balance problems. And one of the things that I was very focused on understanding is what are the other non-gait related problems these HSP patients have. And we found that a significant number of them have bladder problems, and they often don't talk about it. And unless the clinicians ask, this is a personal matter to a lot of patients. So it is an important aspect of their care to address. A lot of patients have pain and spasms. Nearly 40% of our patients were in significant amount of pain. So pain they might complain of, but the bladder issues, if you don't ask, you might not know. And same goes for mood and cognitive functions. So in our complicated cohort of patients, we found that ataxia or difficulty in coordination is a problem for a large number of patients, particularly if they have SPG7 mutations. And the cognitive impairment might not be apparent like in a significant dementia, but the difficulties in carrying out a day-to-day task, organizing simple tasks might be a challenge for some patients, particularly if they have SPG11, SPG4, and some patients with spastin. Now, this is what we have found recently and in the process of publishing our data. And one of the other interesting findings from our cohort is that we find these patients tend to also have what is called optic atrophy. Now, optic atrophy is degeneration or changes in the nerve that supplies the eye. Now, early optic atrophy doesn't cause any visual problems that is known to the patient. You might be able to read the optician's nurse chart. But once the vision is challenged with low light or low contrast, then we begin to find there are some problems in ability to see properly in SPG7 and SPG11 patients in our cohort. So to increase this awareness among clinicians so that they go and look for it is important. Now, why do HSP patients have different types of complications to the others? That's because Hereditary Spastic Paraplegia is a large umbrella term. Within that, there are multiple spectra of diseases. Some patients have spastic paraplegia and ataxia, as you can see. Some of them develop spastic paraplegia and intellectual disability. Some of them have spastic paraplegia and muscle wasting. And you could see that it is not one disease. It is a composite of multiple diseases. So one needs to be aware of this spectrum when you investigate, when you try to look for a genetic cause. I apologize if the gene related to you is not in this diagram. This only goes up to SPG72, but one has to be aware that it is a spectrum of conditions, and one of the genes that is classical of showing this spectrum is SPG7. So SPG7 group of patients is increasingly recognized, and recently we published a large cohort of SPG7 patients in our HSP group, and this is the largest in the UK and to our knowledge the second largest in single center cohort in the Europe. Since our publication, Corelli et al. Published a large number of patients in a multi-center collaboration in Europe, and it's important to draw from our experience. The other observation that we made is that in Sheffield we have the National Ataxia Center, and in that we have a large number of genetic ataxias. 10% of the genetic ataxias are also caused by SPG7 patients. So some of these patients might have ataxia rather than HSP like phenotype, but when you examine them, you will notice that it is difficult to differentiate the two groups. So what is paraplegia or SPG7? So this is a protein that is coded for in the nucleus, which is the command center of a cell, and it travels into the mitochondria. Mitochondria are the powerhouses within a cell. And you can see here, this is a mitochondria. Mitochondria has inner wall and an outer wall. This paraplegic protein sits in the inner wall. And it sits in the inner wall and controls what crosses that membrane in and out, and it controls the quality of things which are crossing it. And you can imagine that if there is a problem of this protein, then it might not do this quality controlling work properly for the mitochondria, which are the powerhouses in the cell. If the powerhouses are not working, if the electricity is not working, then you can't run the functions in the cell. We talked about the long axons needing very fine management of the environment. So if that is affected by lack of power, then you can imagine that these nerves might start degenerating. So it was discovered in 1998, but it's taken such a long time for it to be properly investigated and looked for in terms of what is causing it, what is the phenotype. There is increased awareness of this protein now, and there's a lot of research starting to happen. In our cohort, we have 55 patients, and we find that the spasticity is only one factor. They might have some muscle weakness in the legs, causing walking problems, what we call waddling type gait, and they might have CAE, cerebral ataxia. And about 1/5 of our patients did have a complicated HSP type phenotype, and some of them have spasticity and ataxia together. So and these patients also have a mild slurring of speech. So they have slight slurring of speech, some spasticity in the legs, some difficulty in balancing, and because it didn't really fit a classical HSP phenotype, people didn't recognize that it could also be a patient with hereditary spastic paraplegia. And we found that these patients tend to, I won't bore you with the finer details, I'll crack to the chase here. And here you could see that the mean age of onset of symptoms could be as late as 42 years. That means patients might present later in their lives. They might have had the conditions which people said, okay, maybe you're a bit clumsy and you're a bit slow, but there's nothing to be found. But later on in their life, they start finding falling and difficulty in coordinating. And this might be attributed to various other things, such as knee joint pain, or that some people are told that that's old age, and things were missed. Or some patients present with droopy eyelid and they can't move their eyes, and they were told that they had strokes. But when you actually examine them, take a history and look at their family, you begin to find that these patients actually have hereditary spastic paraplegia form of disease. And more than 90% of them have degeneration or atrophy of the cerebellum. If you scan them, you can see that there are changes in the cerebellum, which is the balance center. So the other things that we found in this group of patients is that the nerve to the eye shows some changes, and we are doing a collaboration with Professor Paola Gonti in Queen's Square in London, and we put together our patients, and we found that over 44% of SPG7 patients can have changes in the back of their eye, and this might not have a direct clinical change impact in the sense they might not have any problems in seeing when from their experience, but when you challenge their vision, there are problems. And we are also hoping to use this as a marker of the disease progression and as a biomarker to predict how a HSP will behave in a person. And the other thing is what we call ophthalmoplegia. That means when a patient with HSP look around, when their eye movements should be normal. But some patients have limitation. They can't look up, and they can't sometimes look to the side unless they turn their head completely. Now, this is called ophthalmoplegia. What is interesting about our patient cohort is that at the very beginning, when you look at our early data, early records, there were very few patients with this condition. But when you look at the follow-up data, they gradually tend to develop this problem. So this is something that clinicians need to be aware of, patients need to be aware of, that it can happen later on. So Patrick Chinnery, his group showed that after about 20 years that some of their patients with mitochondrial diseases developed this eye movement problem. And within that cohort, there were SPG7 patients. And in Sheffield, we have the access to state-of-the-art brain imaging systems. And when we looked at the MRI scans of our patients, we found a characteristic change in the brain of these patients. For example, if you can, I do not know whether you can appreciate, on the panel C here is a scan, brain scan of a normal patient, normal person. And in there, this black area is called the dentate nucleus. And in the SPG7 patients, this black area has become white. So there's a whitish signal suggesting there is a change in the tracts or the cells in that area of the brain. When we did a statistics on that, we found that SPG7 patients have significantly increased signal change in this area called the dentate. You could have a look at patients' brains, and there is pathological change in their brains. So this is a marker that we are studying to see how does this happen and what is going wrong in the brain itself. Now, we thought it was a spinal cord problem, but there are problems in the brain. So what are the changes in the brain that are happening in this group of patients, and as well as hereditary spastic paraplegia patients, is some of the work that we are doing currently. So in Sheffield, we found that a number of patients have various different mutations. Understanding what genes are, what mutation in the gene is important for the clinicians to who are working on this patient group elsewhere in the world and the UK to further their research. So we have published these genetic details, and it is thought that where the mutation in this gene has nothing to do with their clinical picture. But what we did was we went and analyzed all published literature on this gene up to 2018. There were over about 300 patients data published by that date, and we analyzed all the mutations and their clinical picture and found that actually if these patients have a mutation in a part of the protein called peptidase M, this is just a name, and I will show you that this is a schematic diagram of SPG7 protein, and why it is important to understand where the protein, what important functions the protein has, is to be able to draw important conclusions like this. So in this SPG7 protein, this is called the peptidase M1 domain. So if there's a mutation in this area of the protein, and we showed that these patients tend to have a slightly younger onset of age. In fact, it's not slight, it's 12 years. They're younger by 12 years compared to if the mutation was elsewhere. This kind of data is important to be able to inform the patients prognosis and progression of their condition. We found another interesting change was that SPG7, like SPG11, for example, need two abnormal copies, one from the mother, one from the father. And if you have the same spelling mistake in both the copies, then that is called homozygous. Now, those patients had a slightly less worse. In other words, they had a slightly later onset disease. If you had two different types of spelling mistakes, then they tend to have an earlier onset of the disease. So this is also important when we are planning and research and interventions in future, because if you try to find a treatment that will work for a very heterogeneous group of patients, it might not work. The research might fail, like how it has happened in motor neuron disease. So therefore, you need to be able to find ways to homogenize a group, to minimize the variability within a group. So these kind of studies will help us to pick and homogenize a group to see the maximum benefit of a treatment if it comes up in future. The European cohort also found that if the mutation or change in the gene were to cause premature loss of that protein in the cell, we call it loss of function mutation, where the gene or the protein undergoes premature decay. If you have that mutation, then that tends to cause more spasticity. That is what they found in their cohort. So these observations are important to be able to predict and counsel patients. And I move on to what happens in Sheffield. I'll just quickly check what time. Yeah. So in Sheffield, when the patients attend our clinics, we undertake a detailed clinical assessment on each visit. This also includes a complete examination. The spasticity, stiffness is assessed, across all joints. Their balance and coordination is monitored. They undergo visual assessment. We also undertake mood and memory assessments, because we see that this is an important component for HSP patients. And if they haven't had a genetic diagnosis, we undertake genetic testing, and some patients consent to donate their skin biopsy. In addition to these assessments, we also undertake a computerized gait assessment for these patients on each visit that they come to us. And we also offer them the opportunity to take part in neuro-imaging, various types of brain imaging that we undertake in Sheffield. Not only we assess the patients, but we also offer them therapeutic interventions. So we look after them as spasticity and manage that with pharmacological agents. I'm sure that happens everywhere you are seen. And they also have the opportunity to meet the rehabilitation specialists. We are in the process of combining the clinics with the rehabilitation specialist from beginning of autumn. And they're offered Botox and baclofen pumps and functional electrical nerve stimulation if they have problems with their feet. And we have a specialist physiotherapist, and the patients from our region can access them. But if they're outside the region, I liaise directly with the local physiotherapy team to help them. We also have access to hydrotherapy for patients with spasticity in the legs. We have access to a pain management team we work very closely with. And of course, we have a urologist who has specialized in neurological problems of the bladder. So we also obtain bladder care for them. So with all these things, what are we planning to do next? So what we want to do, and what are we doing now, is we have the fortune of having a large cohort of patients in Sheffield, and they are being systematically followed up. This gives us the opportunity to look at what are the biomarkers, clinical, neurological, radiological, and from assessing the gait, to be able to predict what's happening in the natural history of the disease of HSP patients. Are their disease progress? And this is currently what we are analyzing and what we are doing. And we are able to then look, are there differences between SPG4, for example, spastin, and the difference between SPG7 and other genotypes. And we'll be then able to share this data with other colleagues working in other countries to compare and contrast their results with ours. And the hope is we will be having the same methods of assessing the severity, the progression of the disease amongst different centers. So when it comes to monitoring the gait, we know that HSP patients, this is their primary problem. One of the primary problems is walking, and how can assessment of walking inform us of their disease condition, how the disease is progressing. This is important to me because a lot of patients ask me when they come to my clinic, doctor, when will I become wheelchair bound, like the chap who sat outside in your clinic? I don't have answers to give this because no one has done an extensive natural history study. If they ask me, when will I lose the ability to look around like my father has, then I don't have answers to give that because we have a limited follow-up data and natural history data across the world. So this is the gap that we are trying to fill. And these patients are undergoing detailed clinical assessments, and we know HSP is a slowly progressing condition. So even within a year, you might not see a difference. And in my experience, it might take about 1.5 years for a lot of patients to show a significant a measurable deterioration. You might have fluctuations throughout the course, throughout the year, depending on whether the weather is warm or cold, how your mood is, and whether there's an infection or not. But the objective measurement of this disease change is very slow for a lot of the patients, yet very disabling. And we have experimented on a single sensor. Usually gait assessment will include multiple cameras, big laboratory called the gait lab, and pressure pads on the floor. And you can't do this in a busy clinic. So therefore, a lot of patients miss out on this. So what we have proposed is to have a single sensor that patient wears on the back, walk up and down the clinic floor about 10 meters. And it gives us lots of data that is helpful to analyze what's happening in the patient's walking. And it provides nice graphs with different information about how the right leg and the left leg moves in relation to the other. And it also tells us how the trunk moves, whether somebody is swaying from side to side or back to front. And this is just an excerpt of hundreds of data variables that we can generate from one single sensor, rather than asking somebody to walk up and down 10 meters and time it. That's only one assessment, but we are able to generate from one assessment by using a sensor multiple data points to be able to predict what's happening in a patient's walking. This is some data to illustrate this. This is spastic patients that we have assessed by doing a simple 10-meter walk test using a sensor. We can show that there are significant differences when this stand up from sitting position, when they walk the first 10 meters and then come back, and when you look at the total time, there are significant differences in their simple walk that we are able to then analyze. Looking at one abnormal gait is not enough for us. We need to follow this patient up over a period of time and then see what happens to these parameters that we are monitoring. And if somebody then became, for example, unfortunately, wheelchair bound, then we could look back at their data and see what change perhaps would have predicted this happening. And if we collect this over a period of time for a number of patients, then we'll be able to share that information in a model to help advise and counsel other patients. And not only for that, we will then be able to predict if this particular parameter is changing, then we might be able to intervene earlier on, advise them to stretch their ankles, undertake physiotherapy, hydrotherapy, etc. Because there are a lot of young patients I see who are reluctant to maintain and look after their ankles and stretch and take necessary care, mainly because they're strong and they're feeling well in themselves. They can't see what might happen in future if the disease progresses. And this is SPG7 patients. Again, in this cohort, we could see that they have changes in sitting down from standing position compared to spastic patients who had problems with standing up from sitting down position. So likewise, we can, using one sensor, we'll be able to predict a lot of things about their gait. If somebody is significantly affected when they walk, you can see that and say that patient's gait is affected. But we have a large cohort of mildly affected patients. When they walk, no one can really say that they have a problem. Unless you tap their reflexes, scratch their plantars, and take a family history, then they know, okay, this is an affected family member. But when you do the gait assessment, our early data show that the gait assessment can show differences between controls and mildly affected patients. Now, these are very important observations in order to make our group of patients what we call trial ready. That means their clinical phenotype is very deeply studied. We have fine data about their walking, and such cohort will be highly desirable to a researcher who wants to study the effect of a drug, because we know that this group of patients are mild, then the other ones are moderate. And if they want to study only the mild patients because they want to see that the drug can stop their progression, then then these studies and assessments are very helpful. In addition to the gait studies, we also undertake neuroimaging, because gait is something that is affecting the legs, but it doesn't give you an idea as to what's happening to the rest of the body, the rest of the brain, apart from the spinal cord. Neuroimaging gives us access to the brain and the brain stem, which is the connection between the spinal cord and the brain. And we know that more axons, more nerves which are damaged, can cause severe phenotype or severe clinical picture. To study how fast the nerves are being damaged and which parts of the brain are affected, we need neuroimaging, and that's what we are doing currently. So all our patients undergo different types of MRI scanning, and with the help of the HSP Support Group PhD studentship, we'll be able to now undertake some of the cutting edge MRI tractography, and I'll show you some pictures. Here you go. So we are able to generate pictures of the different brain connections from different parts of the brain of the patients. And then once we monitor this over years, we could see if there are changes in these tracks and then correlate that or compare that with their clinical picture. And this will give us information, we might be able to predict if this particular change in the brain is happening, these patients might have increasing difficulties in walking, or they might have problems with their balance, with their cerebellum, and henceforth. And so why are we doing this? As I explained to you earlier, we want to be able to tell the patients how the disease might progress, and also we'll be able to predict certain milestones, and if we are anticipating such changes in their condition, we might be able to intervene early with pharmacological or therapeutic measures. And we are all doing this, but that's great. But some patients ask me, is there a treatment for my HSP? Is there a cure? And I feel inadequate most of the time, because we don't have a treatment, we don't have a cure. And what we're doing now, in addition to the work that I said, was to find a cure for HSP. So what we've done is tap on to our expertise in Citron, or in Sheffield Institute of Translation Neurosciences. We have a well-established setup for drug screening. So in this setup, we are looking for large libraries of drug, what we call repurposing purposes, the well-known established drugs in rescuing neurodegenerative phenotype. This is done for Parkinson's disease, motor neuron disease, Huntington's disease, etc. in our laboratory. So we have a well set up mechanism. And what we're doing is now using hereditary spastic paraplegia patients. So as a start, we've used SPG7 patients, paraplegic patients who have donated their skin samples. This is called an in-cell high content imaging screening. So in this machine, we can look at 384 wells with cells from different patients in one go. And in these wells, we can add treatments, we can challenge the cells, we can stress them and look at their behavior and compare that with controls. Because when you look at a cell from a patient with spastic paraplegia and a normal person, you can't tell the difference to the naked eye. So, but these machines will be able to stress the cells, add components, and generate differences. So it's able to differentiate between how a control patient cell will behave and a patient cell will behave. And I'll show you an example of, and then also this mechanism is able to pick up on 26 different changes within a cell. Now, if you look at a cell, you can see the nucleus, which is the red part, and this the green component is the cell body, and this little net or a mesh-like structure is our mitochondria of the cell, and in this blue area are lysosomes, which are important for packaging the waste as well as good materials within the cell, and this high throughput imaging system can pick up various parts in the cell which can change when they are stressed or when the chemicals are added. So in this very complex diagram, what I'm attempting to show you is that these are patients' cells from SPG7 mutations, and they have been subjected to various stresses, and we have added certain treatments to see whether that will differentiate between patients and controls. So in the graph here is that in gray are the controls, and in blue and green here are the patients. And we can see that there are a number of differences that the system can pick up between the patients and controls. And once these differences are picked up, then we can look whether there are drugs that can rectify this difference. And such a hit would be a potential therapeutic target. And this is the mechanism. This is the work of a BMedSci student who just completed, and I hope to continue this work with the help of the HSP Support Group PhD studentship in next few years to come. So in addition to this work, there are different animal models that we are developing, and Dr. Andy Grierson, he has developed a spastin mouse model and has also recently developed a zebrafish model which has swimming problems due to spastin mutation. So these models are important to test out drugs, as well as to understand mechanisms that are causing problems, which then we can exploit to find target to treat. In addition, we are also undertaking gene therapy programs within Sheffield. Professor Mamouna Azouz is particularly interested and a well-established international scientist who uses viral vectors or viruses to deliver genes to the motor cells, so that if the gene is lost or is malfunctioning, then you can as if restart the cell. So he's particularly interested in SPG15 spasticin and developing methods to deliver the healthy gene into the cells. And once this, we can also use the same model for other HSP related genes, and so this is a space to watch. So what do we do next? And as I said, we are continuing this work with the PhD studentship, and then also plan to apply for further grants to extend the neuro-imaging work. So in summary, in the regional HSP clinic in Sheffield, it caters to a large group of HSP patients who are clinically and genetically well characterized. And there is an advantage of a single center to have a uniform standard of care and assessments of these patients. This will help us accrue longitudinal data over time to inform us of disease progression and identify biomarkers. And there is significant opportunity to take part in clinical research in Sheffield. And we have animal models, cell models, and various other methods to monitor the disease progression, such as MRI assessments and gait assessments. I also would like to mention about Sheffield Children's Hospital, and we've identified a large group of our patients have children, and led by Dr. Tony Hart and Dr. Ming Ong, we are providing a service for younger patients with HSP, and with whom we do a joint clinic. No work is possible without the team. And before that, I would also like to thank all the patients and families who have continuously supported us and been with us and trusted in our service all the way along. And special thanks to Hereditary Spastic Paraplegia Support Group for organizing this meeting, and then also funding our PhD studentship, and Medi Foundation for funding the gene therapy work that Professor Mamouna Azouz is doing, the Neurocare and Ataxia UK. And in addition, this is our team, Professor Shaw, Professor McDermott, and Professor Hajiwasilu, and Professor Nigel Hogarth. They are clinicians, and Professor Nigel Hogarth is a neuroradiologist. Professor Claudia Mazza is a bioengineer who helps us understand the gait biomarkers, Dr. Heather Morty-Boyes works with the cell imaging and drug screening. These are our research fellows who have completed the excellent work. And this is our therapy team who recently won an award for a separate condition, but they are extremely vital for our functioning of the clinic. Many thanks.